Okay, we are going to get into chapter 15 and 16 with these next lectures. This one will pretty much cover all of chapter 15. I'm going to try and go a little bit faster through it just so these videos don't end up being hours long. If it is going too fast, since we're watching on YouTube, you can just click the little settings button at the bottom of the video and adjust the playback speed a little bit slower if you need to. Okay, let me mention here. So chapter 15 is all about the autonomic nervous system. Just to recap, the somatic nervous system is what's consciously perceived or the controlled processes of our body. We have the somatic sensory and the somatic motor, all guarding or guiding our voluntary activities, our skeletal muscle activities. Voluntary movements involve the cerebrum, reflexive movements, and the brain and spinal cord. There's that basic setup of the somatic nervous system that we've seen. The posterior root ganglion and the posterior root receiving those sensory signals, whereas the anterior root is delivering the motor signals to the effectors. In the autonomic nervous system, it's also our visceral motor system. So, the autonomic nervous system has the autonomic motor system, which is regulating the processes below conscious control. It transmits signals from the central nervous system to the heart, smooth muscles and glands, and responds to sensory input from blood vessels and organs. Its primary function is to maintain homeostasis. So whether it is regulating us up or regulating us down, it's trying to maintain homeostasis the whole time. So it looks very similar to the somatic nervous system. We have the sensory signals coming in through the posterior root, motor signals going out through the anterior root to the effector organs. So the lower motor neurons of the somatic nervous system, looking at their structure. So a single lower motor neuron extends from the central nervous system to skeletal muscle fibers. You have the cell bodies within the brainstem or the spinal cord. You have a myelinated axon with large diameter for rapid conduction, and it releases acetylcholine to excite the muscle fiber. So that's our normal somatic nervous system. We've seen this plenty of times. The autonomic nervous system uses the chain of two neurons to reach its effector. Its first one is called the preganglionic neuron. So if you remember, ganglion refers to the cell body. So preganglionic neuron houses its cell body within the brainstem or the spinal cord. It has a thin myelinated axon, so it's going to be a little bit slower because it's thin, that projects into the autonomic ganglion and peripheral nervous system, It releases acetylcholine from the synaptic knob to excite a second motor neuron. So in your ANS chain, you have two motor neurons. The first one is going to be the preganglionic. It's going to release acetylcholine to excite the second neuron. The second neuron is going to have its cell body within the autonomic ganglion. So we'll see that the ganglion is just the chain that it's going to be housed in. It's very thin and unmyelinated, so it's going to conduct slowly its signals. It's going to project into the cardiac muscle, smooth muscle, or glands. And the reason that it has a thin unmyelinated chain is we don't want quick, rapid changes. We want these changes to be slower and gradual so they can be monitored and regulated more precisely. The second neuron in this autonomic nervous system chain is going to release acetylcholine or norepinephrine to either excite or inhibit the effector, just depending on what type of receptor the effector has. So the preganglionic is going to release acetylcholine. It is going to be excitatory. The second neuron, the preganglionic one. The postganglionic one is going to release norepinephrine or acetylcholine and can be excitatory or inhibitory. This is that essential structure of the lower motor neurons for them. You have this preganglionic cell body within the spinal cord, this axon for the preganglion. You have the ganglionic neural cell, the autonomic ganglion, where it synapses with this postganglionic neuron that then goes to the effector. So your autonomic nervous system, your motor neurons for those. So what advantages are there for the ANS to use a chain of two neurons instead of just one neuron going directly, synapsing directly to the effector? Two things. It allows for neural convergence and neural divergence. So neural convergence is when multiple preganglionic neurons synapse on one ganglionic neuron. So multiple preganglionic going onto one ganglionic, so that way it can bring a lot of different signals to help really regulate a specific signal based on different inputs. As well as neuronal divergence, where you have branches of the axon from one preganglionic neuron synapsing with numerous ganglionic neurons. So one preganglionic synapsing with multiple ganglionic ones just allows for a divergence of that signal, so you can spread your signal out across a broader range if you need to. So the neuronal convergence allows for a lot of input for one regulated signal, whereas neuronal divergence allows that one regulated signal to then be spread out across a larger field of effectors. So how is the ANS regulated? The ANS is regulated by parts of the brainstem and the spinal cord. You have your hypothalamus is crucial in regulating the ANS. It contains nuclei controlling both divisions. We'll see that the fight or flight and the rest and digest systems. The hypothalamus is essential for emotional ANS processes such as our fight or flight, our sympathetic nervous system reactions. It can be influenced by areas of the cortex, the thalamus, and the limbic system. So our emotions do influence our hormones and our nerves. The brainstem nuclei mediate our visceral reflexes. So remember, the nuclei are the gray matter, the cell bodies and the processing centers within the brain. So within the brainstem nuclei, where they are mediating those visceral reflexes, maintaining blood pressure, movement of food through our GI tract, those types of things. And then the spinal cord. It governs some of our autonomic nervous system reflexes, like defecation, urination, are governed within the spinal cord. So here is just a little picture breakdown. I think this was in one of the worksheets. You have your hypothalamus, your brainstem, and your spinal cord, each regulating the ANS in a different way. So the two different divisions of the autonomic nervous system. We have the parasympathetic, which is our rest and digest, and the sympathetic, which is the fight or flight. They have complementary functions. It doesn't mean that every organ, every effector is going to have both a parasympathetic and a sympathetic innervation. We'll see that some only have sympathetic and some have stronger innervations from one division than the other. But essentially you have within your autonomic nervous system, you have your parasympathetic and your sympathetic. Your parasympathetic is what's going to be normally bringing your body back down to a level that is the rest and digest. So you're conserving energy and replenishing nutrients, whereas your sympathetic is going to be more for fight or flight, exercise, excitement, and emergency. So your parasympathetic is your craniosacral division. The preganglionic neurons in the brainstem or S2, S4 spinal cord. So either really high up or really low down. Your preganglionic axons are long and the postganglionic axons are short. We'll see what that looks like in a second. And the ganglia are close to or within the effector organ. Sorry, I have another dog who is drinking water now. I hope that's not too loud for y'all. Okay. The ganglia are, so the cell bodies are close to or within the effector organ. So they're going to be I hope that y'all can hear me over that dog drinking water. So they're going to be able to synapse very close to it. Preganglionic axons have few branches, so there's not going to be a lot of divergence from the preganglionic parasympathetic neurons. In the sympathetic system, we have the thoracolumbar division, so most of the neurons, cell bodies are going to be arising from that thoracolumbar portion of the spine. You have preganglionic neurons and lateral horns of the spinal cord. The axons, the preganglionic axons are short, whereas the postganglionic axons are long, so the opposite of the parasympathetic. And your ganglia are close to your spinal cord, lateral or anterior to it. We'll see that there are two different locations for the sympathetic neuron ganglia. So as opposed to being closer to your effector organ, the sympathetic axons are closer to the spinal cord, and the preganglionic axons have many branches, so a lot of neuronal divergence in the sympathetic compared to almost none in the parasympathetic. So here are the two divisions of the autonomic motor nervous system. You have this parasympathetic, again, it's originating in the brainstem and the S2, S4 sections of the spinal cord. So up here superiorly and then lower down here inferiorly, whereas the sympathetic division has the T1, L2 segments of the spinal cord. We'll get into some of these nerves specifically, but the parasympathetic division uses cranial nerves 3, 7, 9, and 10. The vagus nerve we'll see is very important for this, as well as the pelvic splanchnic nerves. The sympathetic division, it has what's called the sympathetic trunk, which again, we were saying that cell bodies of the sympathetic nervous system, as opposed to being within the brainstem of the spinal cord, are just lateral or anterior to it. So this trunk is where the actual cell bodies, the ganglia of the sympathetic nervous system are housed. We'll see it's called the sympathetic trunk ganglion. And you can see the structural differences between them too. You have your pregangliotic neuron with this long axon, this autonomic ganglion that's going to be closer to the effector organ, and then a short postganglionic axon to actually go into the organ and affect its response, versus sympathetic with this short preganglionic neuron that's highly branched, and then a longer postganglionic axon. Okay, parasympathetic activity tends to be fairly localized. The long preganglionic axons with the limited branches influence only one or a few effectors at the same time, whereas a sympathetic activity tends to facilitate mass activation. You have a short preganglionic axons with many branches simultaneously influencing several effectors, so its response is being spread out. Exercise or fear can stimulate changes in organ systems that can cause different innervations to occur. Sympathetic stimulation of the release of adrenal medulla hormones facilitates this. So we'll talk about norepinephrine and epinephrine are adrenaline hormones coming from the adrenal medulla. And sometimes a single effector can be engaged. So instead of causing this mass activation, it can cause just a response in a localized spot, such as triggering pupil dilation. So parasympathetic division is maintaining homeostasis while we're at rest. Its ganglia are found either in the terminal ganglia, close to the effector, so terminal, close to the effector, terminal end effector, or the intramural ganglia located within the wall of the target organ. So intramural, within the wall of that target organ. So your ganglia for your parasympathetic, intramural or terminal. The nerves that the cranial nerves rather that we use for our parasympathetic nervous system, we said are the oculomotor nerve. So oculomotor, movements of the eye. It's going to control the ciliary muscles and pupil constriction. The facial nerve is going to control lacrimal glands and small glands of the nose and the mouth, as well as submandibular and sublingual salivary glands. When you do 169 and you get digestion, you'll talk more about the salivary glands. But submandibular under the mandible and sublingual under the tongue. Your glossopharyngeal nerve, it's going to have axons controlling the parotid salivary glands. Those are the big ones on the side of your face. Your vagus nerve, this is again, like we said, a very important nerve in regulating our body systems in the autonomic nervous system. It has a variety of ganglia in the thorax and the abdomen, slowing the heart rate, constricting lung bronchioles, increasing digestive secretions, storing carbohydrates. You can see in just these few examples a wide variety of functions. The pelvic splanchnic nerves are going to contribute into the superior and inferior hypergastric plexuses. Just like with arteries and veins, a plexus is a grouping of nerves. So you have your superior and inferior hypogastric, so hypogastric below the stomach, and abdominal and pelvic effectors. They're going to go to abdominal and pelvic effectors. Smooth muscle contraction in the GI tract, increased secretions in urinary and digestive systems, penile and clitoral erections. So pretty much all of our lower abdominal organs. Here is just an overview of this parasympathetic pathways. So you can see these cranial nerves going out to their effectors, this oculomotor nerve. You have your vagus nerve here. Again, it has these several plexuses. So the cardiac plexus is a group of nerves that's going to be going in towards the heart. Your pulmonary and esophageal plexuses, the abdominal aortic plexuses, going to all of these different organs. Once again, remember this is the parasympathetic system, so you see we're arising from the brainstem and S2, S4. Nothing is coming from the thoracolumbar or even cervical regions. We'll talk about this in a little bit, but it is we'll talk about the differences between sympathetic and parasympathetic stimulation, whether they cause vasoconstriction or vasodilation or bronchoconstriction, dilation. There will be some specific examples that you'll need to know. So sympathetic function is related to exercise and emergency. The ganglia, the cell bodies, are close to the central nervous system, but anatomical pathways are complex. So getting to the effectors are more complex. You have sympathetic preganglionic neurons with their cell bodies in the lateral horns and those C1L2 segment. They have myelinated axons that course through the anterior roots of those spinal nerves and then branch off. So myelinated axons increases the speed through the anterior roots rather than the posterior roots, because the anterior roots are carrying motor signals. There are things called sympathetic trunks and ganglia. So there are left and right trunks just lateral to the vertebral column. So on either side of the vertebral column, there are those trunks that resemble a pearl necklace. I pointed them out on that other side. You have a string that are the axons connecting the different pearls, which are the ganglia of the sympathetic cell bodies. Okay, so as opposed to being housed like the parasympathetic nervous system within the spinal cord, these are going to be housed just outside of the spinal cord and create the sympathetic chain. Usually there's one ganglion associated with each spinal nerve, so one ganglionic cell body associated with each of the spinal nerves. But the cervical portion of the trunk has only three: the superior, the middle, and the inferior. So here for your spinal pathways, you have just to the side of the spinal cord the sympathetic trunk ganglia. Sympathetic trunk ganglia. You see from here you have this short preganglionic axon from spinal cord to ganglion to the trunk, and then you have a longer postganglionic axon going from the sympathetic trunk to the effector organ, whether that be the eyes, the heart, the skin, the uterus, whatever it may be. Here's an anatomical example of that. So this is your aorta, this is a vein, these are vertebrae right here. You can see each of the vertebral bodies on there. This right here, this kind of thing that's going down and has little branches, this is your sympathetic trunk ganglion. So the entire sympathetic trunk. Each of these kind of clusters, these thicker clusters, those are the ganglion, the cell bodies. And then these thin lines right here connecting them, those are the trunk. That's the actual trunk. That's the axon connecting the cell bodies. Okay, so you have white and gray rami that connect the spinal nerves to the sympathetic trunk. So going from this sympathetic trunk, you have your spinal nerves over here. These are intercostal nerves, these kind of thicker ones. You have your sympathetic trunk, and then within the vertebrae, you have your spinal cord. So the white rami, remember they're white, so they're going to be myelinated, are going to carry signals from the T1, L2 nerves to the trunk. So they're going to go from the spinal cord, from the spinal cord to the trunk ganglion. The gray ones are gray, so they are unmyelinated, and they're going to carry axons from the trunk to the spinal nerves. So they're an exit ramp from the trunk. So this is going from the spinal cord. White rami go from the spinal cord to the sympathetic trunk ganglion, whereas the gray rami go from the spinal nerves back towards the spinal cord. They have to just go through the trunk to get there. So you can see this gray one right here is much, much thinner. So signals coming from the spinal nerves to the trunk to the spinal cord are conducted on thinner and unmyelinated neurons or axons, whereas signals going from the spinal cord out to the spinal nerves are going to be in thicker myelinated ones. Okay, your sympathetic splanchnic nerves, I think that's how you say that. You have preganglionic sympathetic axons not synapsing in the synaptic trunk, or rather in the sympathetic trunk. Rather instead of being in that sympathetic trunk ganglion, they're going to run anterior from that and typically terminate in the prevertebral ganglia. The prevertebral ganglia are located anterior to the vertebral column on the surface of the aorta within that abdominal pelvic cavity, and there are several of them. So this is just a different place for it to synapse. So as opposed to synapsing in the trunk, it's going to synapse in the prevertebral ganglia. Your celiac ganglia innervate the stomach, duodenum, spleen, liver, gallbladder, and pancreas. Your superior mesenteric innervate the small intestine, large intestine, pancreas, kidneys, and ureters. So you can see all of these things are in our abdomen. Your inferior mesenteric innervate the large intestine, rectum, bladder, ureters, and reproductive organs. So what we're looking at is these sympathetic splanchnic nerves are going to go not from the sympathetic trunk, they're going to go into these prevertebral ganglia that will then innervate abdominal pelvic organs. So there are different pathways. Axons exit the sympathetic trunk by one of four pathways. So once they go from the spinal cord to the trunk, they have to then go out some way. The first one is the spinal nerve pathway. This is for skin effectors of the neck, torso, and limbs. You have the postganglionic axon traveling through the gray ramus, joining that level spinal nerve and extending to its effector. I think that's all that I wanted you to know. Yeah. So you can see that it goes out through this anterior horn, it goes into the ganglionic trunk, and then it synapses to go to its effector. kind of a very standard pathway of what you would have expected considering what we've talked about. That's how it would go. It's not the only way that it can go though. The postganglionic sympathetic nerve pathway are for effectors that are internal organs of the thorax and neck, skin effectors of the head, neck, and eye, or head and neck, and eye and eyelid and eyelid or pupillae muscles. These are going to have preganglionic neurons that synapse in the sympathetic trunk ganglion. So these are going to go to that sympathetic trunk, but then they're going to go from the trunk directly to the effector. It doesn't go out through the gray ramus. So these we're exiting out through the gray ramus, I believe, and these are not. So you can see it goes out through here, and then it goes to the neuron and goes right to the effector. Your splanchnic nerve pathway is for effectors in the abdominal and pelvic viscera. You have preganglionic axons that pass the sympathetic trunk without synapsing, so they don't synapse in that sympathetic trunk. Remember, these are the splanchnic nerves. They're going to go into that prevertebral ganglia where they synapse. So they go to that prevertebral ganglia as opposed to in the sympathetic trunk. You have your adrenal medulla pathway, which is for the central region of the adrenal gland. The medulla is the medulla middle of the adrenal gland. You have preganglionic sympathetic axons extending through the sympathetic trunk and prevertebral ganglia without synapsing in either. So this does not synapse in the sympathetic trunk or the prevertebral ganglia. The preganglionic cells stimulate adrenal medulla cells to release epinephrine and norepinephrine into the blood, enhancing or prolonging the fight or flight response, the sympathetic nervous system response. So these exit out, and then they innervate directly into the adrenal medulla. So these autonomic plexuses are networks of nerves within the thorax and abdominopelvic cavities. They contain sympathetic postganglionic axons, so sympathetic postganglionic, parasympathetic preganglionic, and some visceral sensory axons as well. There is the cardiac plexus. What I want you to know is you should be able to label or identify each of the different plexuses and what the sympathetic and parasympathetic activity is going to do. So cardiac plexus, its sympathetic axons are going to quicken the heart rate. Parasympathetic axons from the vagus nerve will slow the heart rate. The pulmonary plexus, sympathetic axons, bronchodilation. Parasympathetic axons, again, vagus nerve, bronchoconstriction. Esophageal plexus, sympathetic axons will decrease motility. Parasympathetic axons coordinate opening of the cardiac sphincter with swallowing, so actually allowing motility. The abdominal aortic plexus, it's consisted of these minor ones. You should be able to recognize that. Yeah, if you see something celiac, mesenteric, you should automatically start thinking that is in your abdomen. When you do cardiovascular system, chapters 18, 17, 18, 19 stuff, or 18 and 19, you'll get into more of these celiac and mesenteric vessels. So just every time you see celiac or mesenteric, think that it's in your abdomen. The sympathetic axons, innervate all abdominal and some pelvic organs. Your hypogastric plexus innervates viscera within the pelvic region. Okay, so there is just an overview with the different plexuses, the cardiac, pulmonary, esophageal, abdominal aortic, hypogastric, and some of the nerves labeled. Your enteric nervous system. This is essentially the part of your nervous system that extends into your gastrointestinal system. Its autonomic motor and visceral sensory neurons arranged throughout the wall of the GI tract. It works to innervate smooth muscles and glands. It coordinates peristalsis. And it has two large plexuses, the submucosal and the myenteric plexus. So the submucosal plexus is part of the mucosal layer of the GI tract wall, and that one is going to help release secretions from glands, because essentially what glands do to get them to release their secretions, it's not that they get like kind of squeezed and stuff oozes out. These submucosal plexus will innervate little muscles around the glands that actually makes them kind of jiggle, and when they jiggle, that's when they release their stuff. So you have these submucosal plexuses that are going to help release from glands, and the myenteric plexus myo. So this is the muscle layer of that smooth muscle of the GI tract wall. Okay, neurotransmitters of the autonomic nervous system. We use both acetylcholine and norepinephrine. Either neurotransmitter can be stimulatory or inhibitory. It depends on your postsynaptic receptor. That's what's the important thing here. So ACH or norepinephrine, epinephrine, they can both be excitatory or inhibitory. Just depends on that postsynaptic receptor. Cells that release acetylcholine are called cholinergic neurons. So if it releases acetylcholine, it's a cholinergic neuron. And target cells will have cholinergic receptors. Makes sense. All your cholinergic neurons are going to be all preganglionic neurons, all parasympathetic ganglionic neurons, and sympathetic ganglionic neurons innervating specifically your sweat and blood vessels and your in your skeletal muscle. Okay, so these are going to be the cholinergic neurons that we're going to mostly see. Cells that release norepinephrine are called adrenergic neurons. Most of your sympathetic ganglionic neurons are going to be adrenergic, and the target cells have adrenergic receptors. These are fun words to say. Okay, so you have acetylcholine coming from cholinergic receptors, norepinephrine, epinephrine coming from adrenergic receptors. Your cholinergic receptors, all of your preganglionic neurons, so preganglionic, whether they are the sympathetic or parasympathetic, all of your parasympathetic ganglionic neurons, so basically your postganglionic parasympathetic neurons, and your sympathetic ganglionic neurons going specifically to sweat glands and blood vessels in the skeletal muscle. So here is just a picture summary of that. You have your parasympathetic pathways. Again, all of the preganglionic ones are going to release acetylcholine. Okay, so you can see that all the preganglionic ones release acetylcholine. It's in the postganglionic that things start to get a little bit different. So our parasympathetic, it's still going to be releasing acetylcholine. It's because all parasympathetic are cholinergic. Your sympathetic, they can be cholinergic or adrenergic, right? So you can either have acetylcholine or norepinephrine being released. It just depends on what the target cell is. So it's a cholinergic receptor because it's releasing to the target cells in the sweat glands. Most of the body structures, though, it's going to be an adrenergic response. Within the adrenal medulla cell, you can see, remember that it's the sympathetic pathways that we talked about just before this. We said that the sympathetic neuron is going to go directly to the adrenal medulla. It's going to pass the sympathetic trunk. It's going to pass the prevertebral ganglia, and will go directly to the adrenal medulla, where it will also release acetylcholine. It will also be cholinergic. So two main types of cholinergic receptors. We'll see that there are also two types of adrenergic receptors. So cholinergic receptors are going to be binding acetylcholine. You can have either a nicotinic or a muscarinic receptor, which you may have already heard these words before in some of the other things we've talked about. So your nicotinic receptors are found on all gangliotic neurons and the adrenal medulla cells. They're also on skeletal muscle cells at the neuromuscular junction. So if you have trouble kind of remembering which ones bind to what, if you can remember that nicotinic are on skeletal muscle cells, and you remember that to have skeletal muscle contraction, you have to have acetylcholine, that might be able to help you connect, okay, so nicotinic are binding acetylcholine, skeletal muscle cells have nicotinic receptors. It helps me. When acetylcholine binds a nicotinic receptor, it opens cation channels, so sodium is moving out. Sodium moves into the cell and the cell depolarizes. Again, we know this, we've done muscle contraction. It gives you a excitatory postsynaptic potential for hopefully a full depolarization if that's what's needed. Subtypes of nicotinic receptors react differently to drugs depending on which organ they are located and which organ they are located. So location is a big important thing. Just because it has the same receptor as a skeletal muscle cell doesn't mean that it's going to respond the same if you are in your liver. Your muscarinic, so that was the nicotinic. The muscarinic, the second subtype of our cholinergic receptors, found in all target organs of parasympathetic division and a few of the sympathetic divisions, so these are a little bit more widespread. You have sympathetic effectors with muscarinic receptors, include your sweat glands and blood vessels in the skeletal muscle, obviously because that's where the sympathetic effectors actually release acetylcholine, is in these two regions. All muscarinic receptors use secondary messengers. but different subtypes of receptors have different effects. So they're going to have those G protein second messenger systems, cascades. When acetylcholine binds to muscarinic receptors of smooth muscle in the GI tract, it's stimulated to contract more. So you have your acetylcholine binding in the GI tract, contracts. Acetylcholine binding receptors in cardiac muscle will cause the heart rate to decrease. So again, it's not always going to be causing the same effect just because they're both binding muscarinic receptors. The organ, the effector organ does make a difference. Okay, adrenergic receptors. Norepinephrine, and again, when we say norepinephrine, you can also put in epinephrine. They are very, very similar. Norepinephrine has a little bit stronger effect on blood vessels, epinephrine more so on your heart, but they interact and act the same way. So norepinephrine is a catecholamine. Catecholamines are a specific subtype of biogenic amines that will bind to membrane receptors. So remember, there are ligands. Ligands are things that bind to cells, receptors on different cells. Your norepinephrine and epinephrine bind to your adrenergic receptors. Okay, so norepinephrine and epinephrine are catecholamines. Catecholamines are biogenic amines. These norepinephrine's can also bind to adrenergic receptors. Two main types of adrenergic receptors. We had two cholinergic, we have two adrenergic. The adrenergic receptors are alpha and beta. Cells with alpha receptors are typically stimulated by norepinephrine, whereas cells with beta receptors are stimulated or inhibited by it. Okay. So it's a little bit more with the adrenergic, sorry. The cholinergic, you had nicotinic, muscarinic, and that was pretty much it. With adrenergic, you have alpha 1 and alpha 2. So the alpha 1 are stimulatory receptors found on most smooth muscle cells. They do vasoconstriction, goosebumps, uterine contractions, contracting the ureters, close off the internal urethral sphincter, and pupil dilation, so that which is constricting it back. The alpha 2 receptors are located throughout the CNS and the pancreas, inhibiting insulin secretion, and the GI tract sphincters, causing constriction there. Okay, so you have alpha receptors. These are just different subtypes of them. Your beta receptors, there are 3 subtypes. I've never heard of the beta 3 subtype until I started reading these notes. Beta 1 and 2 are the primary ones that you see. The beta 1 are mostly stimulatory, located in your heart, increasing heart rate and force, and in the kidney to stimulate renin secretion, which will also, you'll learn renin stuff in 169, but it increases blood pressure. Your beta 2 receptors are primarily inhibitory, so when they release their signal, or when they bind to epinephrine or norepinephrine, that's going to cause an inhibitory response. They're located in the smooth muscle of the vessels to the heart, liver, and skeletal muscle for vessel dilation, the lung and bronchodilation, uterine and GI tract smooth muscle cause relaxation, and the detrusor muscle of the bladder causes it to relax as well, so it can fill with urine. If you may have heard of people taking beta blockers, these are to help lower your heart rate. So your heart has both those has these adrenergic receptors that can be blocked, so that way your heart rate can slow down if you have too high blood pressure. Okay, autonomic tone. Most effectors are innervated by both divisions of the autonomic nervous system. They're both going to be continuously releasing neurotransmitter to generate this autonomic tone and to keep us, again, in that homeostatic balance, that even place. In some instances, only one division will control the status of an effector. Blood vessels are usually due to sympathetic tone, either more or less sympathetic tone, specifically called your vasomotor tone. If sympathetic activity increases, you have vasoconstriction, vasodilation if sympathetic activity decreases. Your vagal tone as well continuously decreases your heart rate. Your heart rate would be about 20 beats higher a minute if without continuous inhibition from the vagus nerve. We can have dual innervation, where an organ receives input from both the sympathetic and parasympathetic divisions. That can be either antagonistic or cooperative. Antagonistic, usually the two divisions oppose each other. You can see in the parasympathetic activity will slow the heart rate, sympathetic activity increases heart rate. Your cardiac cells, as we said, have both cholinergic and adrenergic, so they can have these competing signals to kind of find that optimal heart rate for the moment, because it will change a little bit. You can have muscular activity in your GI tract. Parasympathetic activity will increase GI tract motility, whereas sympathetic will decrease it. Your GI smooth muscle cells have both types of those receptors. They will obviously be able to get innervation from both. And as well as pupil diameter of the iris, the parasympathetic activity will decrease your pupil diameter, whereas your sympathetic activity dilates your pupil. Cooperative effects, how do they work together? So it's seen when your parasympathetic and sympathetic stimulation have different effects that are part of an overall response, so still working towards the same goal. One of the big ones, male sexual function. So the penis erection is due to parasympathetic activity, ejaculation is due to sympathetic activity. One of a kind of like a little childish way almost to remember that is point and shoot. So the erection is due to parasympathetic activity, so point for parasympathetic, and ejaculation, shoot for sympathetic. That's what I got. All right, opposing effects can be achieved without dual innervation. Your blood vessels can constrict with increased activity, dilate with decreased sympathetic activity. We did that. And other glands that are only innervated by the sympathetic system. Your sweat glands in your trunk, the erector pili muscles in the skin, and your adrenal medulla neurosecretory cells. Neurosecretory cells, epinephrine and norepinephrine. All right, autonomic reflexes, your visceral reflexes, are pre-programmed responses to a stimulus generated by a reflex arc. We did reflexes, what was it, chapter 13, I think. So this should just be a little bit of a review. This is specifically in the autonomic nervous system. You still have a receptor that's going to be detecting the stimulus, sensory relay to your central nervous system, CNS integration, motor neuron, and then an effector. All ANS effectors are cardiac muscle cells, smooth muscle cells, or glands. Your cardiovascular reflex is to maintain blood pressure. Stretch receptors in the vessel wall respond to pressure elevation. So as your blood pressure increases, the vessel walls are going to be stretched, and the receptors within the vessel walls will detect that stretch. They send signals to your cardiac center and your medulla oblongata, part of your brainstem, to inhibit sympathetic and activate parasympathetic output to the heart. So this is going to help to inhibit sympathetic and activate parasympathetic. So it's going to slow your heart rate down and decrease the volume of blood ejected, decreasing your overall blood pressure. This is just one thing. Blood pressure has a lot of different effector responses. Your gastrointestinal reflex is for defecation. You have sensory neurons in the rectum respond to stretch by fecal matter. Again, same thing. As the fecal matter moves into the rectum, it stretches, it puts pressure on those mechanoreceptors, and they send signals to the spinal cord. The spinal cord signals are sent through the motor neurons to stimulate the rectum to contract and the internal anal sphincter to relax. We have two anal sphincters. You have an internal one that is involuntary and your external one, which is voluntary. So once we have this gastrointestinal reflex, the internal one will relax, and then we control at that point when we defecate. So notice this is going through the spinal cord. It's not going all the way up to the brainstem. It's just going to be staying in the spinal cord. Your micturition reflex is your urination reflex. Again, stretch receptors signal when the bladder fills with urine. You get a couple of warnings. You get 1 or 2 warnings before your bladder really is full and the pressure in it is so much that it just you use the restroom on yourself. The signals are sent to the sacral spinal cord, so very low down in the spinal cord. In infants, this results in reflexive contraction of smooth muscles in the bladder and relaxation of urinary sphincters because they can't control that external urinary sphincter. Just like we had an external anal sphincter, we have an internal and external urinary sphincters. The signal here will, the autonomic signal will release the internal urinary sphincter, and then as we get older, we get more control in our pons to control that external urinary sphincter as well. And there's just an image of your micturition reflex. And I think that's all for chapter 15.